morning. Good morning, ma'am. Good morning, everyone. I am Shivani from BTC Combination, and I am here today to present you a seminar on the topic of use of genetic engineering in the development of therapeutics and vaccines, production of biopharmaceuticals using recombinant DNA technologies. So, starting with the class, this is about how we use genetic engineering for the development of therapeutics and vaccines and biopharmaceuticals. Can anyone say what is genetic engineering? Define genetic engineering. Even in simple terms, why do we use it? Genetic engineering is described, it's a modification of organism genetic material using biotechnology. Involves techniques like recombinant DNA technologies, CRISPR-Cas9 and gene clonings to alter genes for desired reason functions. In medicine, it's used for production of therapeutic proteins such as hormones, enzymes, and also used in gene therapies. Vaccines for curing infectious diseases. So anyone say what is genetic engineering? Why is it used? Modification of So first of all, what are therapeutics? These are biologically derived treatments such as proteins, antibiotics, and nucleic acids. Engineered using genetic engineering for treating diseases and preventing diseases and for improving health. Please write down what are therapeutics. These are given for 2 marks in the first period.
You're running with your can you please move on?
Yes. So these therapeutics are often used in living systems like bacteria, yeast, or mammal cells through methods such as recombinant DNA technologies, making them more targeted, effective, and safer than traditional drugs. So these therapeutics are mainly used, are genetically modified, mainly to prevent diseases, to treat diseases, and improve one's health. Examples such as insulins, please note down. Insulins, growth hormones, interferons, monoclonal antibodies, and gene therapy. This is a picture of gene therapy where we have naked DNA plasmid, liposome. These are all vectors and non-vector mediated gene transfers used in gene therapy to prevent diseases or to cure the disease. Example for therapeutics, insulin, produced using genetically modified E. coli bacteria, used for treating diabetes, replace animal-derived insulin, improving safety and compatibility. Before gene therapy or therapeutics, we used pig insulin. So after taking its liver tissues and extracting insulin, they had certain drawbacks such as it wasn't compatible with human insulin, and it has many impurities which made the therapeutics a negative thing. So using genetic engineering, that problem was solved. The next one is erythropoietin, produced in CHO cells. CHO means Chinese hamster ovaries. These are the standard industrial cells. Chinese hamster ovaries are essentially used because due to their identical translation process in translational modifications. That's why these CHO, Chinese hamster ovaries, are most used in producing. These are used to stimulate blood cell productions, used to treat anemia and in chronic liver disease, chronic kidney diseases and cancer patients. Human growth hormones, produced using recombinant bacteria, treats growth disorders in children and adults. Then comes monoclonal antibodies, engineered to target specific antigens, HER2 in breast cancer. Examples include hepticin and pneumonia, mainly used for treating cancer. cells. These are the types of cancers. So these are cured using monoclonal antibodies. Next comes gene therapy, also given in 2 months main and in 5 months questions. Define gene therapy. So please note down. Gene therapy involves the introduction and removal or alteration of genetic material within a person's cell to cure the disease. The question was what define genetic engineering and write its role in preventing genetic disorders. So you can write the whole slide for 5 marks. Monogenic disorders, it can be 3 diseases caused by mutation in a single gene such as cystic fibrosis, sickle cell anemia and spinal muscular atrophy are prime condition for gene therapy. By replacing and repairing a faulty gene, symptoms can be ameliorated or eliminated. Blood disorders such as beta thalassemia and hemophilia can be treated using CRISPR-Cas9 and other gene editing tools. Technologies deriving gene therapy. Modern gene therapy relies on several cutting-edge genetic engineered tools such as CRISPR-Cas9, a precise genome editing tool that can cut and modify DNA and targeted locations. Can anyone say what is elaborate CRISPR-Cas9? And viral vectors, modified virus like adeno-associated virus or lentivirus are commonly used to deliver gene therapeutics genes into the cell. Next comes vaccines. These are not asked, but it's there. Vaccines are biological preparations that train your immune system to recognize and fight specific diseases without causing the illness itself. Means these are inserted into the body. It fights vaccine but doesn't induce infections in the body itself. Can anyone name a vaccine? We used it mainly during COVID. So genetic engineering allows scientists to precisely design vaccines by inserting genes that encodes disease-specific antigens into the host cell or delivery system. These engineered components then stimulate the immune system without using live or whole pathogens. Types of genetically engineered vaccines includes recombinant protein vaccines, use of genetically engineered organisms like bacteria to produce viral proteins. Example, hepatitis B. Vaccines use yeast to produce the surface antigen of the virus. Hepatitis B is the case where your liver just expands. The inflammation occurs in the liver, so that kind of condition is known as hepatitis B. So recombinant protein vaccines are used to treat this disease. DNA vaccines contain plasmid DNA with the DNA encoding the antigen. Host cell produces the antigen after the uptake of DNA. Example, Z-CoV-D, India's DNA-based COVID-19 vaccine. mRNA vaccine use synthetic messenger RNA to instruct cell to produce the antigen. Example, preferred biotech and Moderna COVID-19 vaccines use mRNA to produce the spike protein of SARS-CoV-2, also known as COVID-19. Viral vector vaccines use harmless virus like adenovirus to deliver genetic material. Example again, Oxford Z COVID-19 vaccine use a chimpanzee adenovirus. The alternative name for this is Covishield. These are the edible vaccines. This particular example is for hepatitis B, which uses corn, potato, lettuce, tomato, and this is a spice. So these are the examples for edible vaccines, which we can directly consume plant edible vaccines. We know the advantages of genetic engineering in the therapeutics and vaccines. So what are the disadvantages of it? Unpredictable long-term effects. Genetically modifications may have unseen consequences on human body or microbial environment ecosystem. There's a concern about the stability of the inserted gene and whether they may mutate or recombine unpredictable. Immune reactions and allergies. Engineered proteins or antigens can trigger unexpected immune response or allergic reactions in some individuals. The risks are especially relevant in vaccine where novel compounds are introduced into the body. So, a person having these vaccines can develop allergic reactions. So, these are the disadvantages. Environmental risks, like genetically engineered vaccines may transfer genes to indigenous microbes, potentially altering ecosystems. Resistance and evolutionary pressure. Pathogens may evolve resistant to engineered vaccines or therapeutics, similar to antibiotic resistance. This could lead to new strains that are harder to treat and or control. Regulatory and safety oversights. Ensuring safety requires rigorous testing and long-term monitoring, which can be difficult and costly. Production of biopharmaceuticals using recombinant DNA technology. Here we are going to discuss about the production of insulin, how insulin is produced. Can anyone just describe the general steps in genetic engineering, like isolation of gene of interest from anyone? Is there? You have already written it. No one. So the first thing we do is isolate the gene of interest. After isolating the gene of interest, we select the suitable vector for it. Suitable vector which has multiple cloning sites where the gene inserted is stable, and suitable host where the vector is stable and can replicate itself without degradation, and then multiplication using bioreactors, fermenters, and then gaining our product. So the production of biopharmaceuticals using recombinant DNA technology involves inserting a gene for a therapeutic protein into a host organism, which can then act as a biological factory to produce large quantities of the desired protein. The process of producing a biopharmaceutical like recombinant human insulin is a classic example that illustrates the key steps. Gene isolation and vector construction. The gene of the desired protein, example human insulin, is isolated or synthesized in a lab. This gene is then inserted into a cloning vector, which is small circular DNA molecule, often a bacterial plasmid. The bacterial vector is engineered that it contains the gene of interest, that is insulin gene, into suitable markers such as antibiotic resistance place and a strong promoter to ensure the gene is highly expressed. Transformation. The recombinant vector is introduced into a suitable host organism for insulin. This is typically a bacteria like E. coli or yeast. The hosts are now transformed and now can carry the replication in the foreign medium. Fermentation and expression. The transformed host cells are grown in large scale in fermented times under controlled conditions. And as the cell multiplies, they read the foreign genes instruction and begin producing the therapeutic protein. For complex protein, host systems like yeast or mammal cells are preferred because they perform post-translation modifications that are necessary for the proteins to be functional. Purification and formulation. After the protein is produced, it is harvested from the host cell. It is a critical step as the protein must be isolated from the host's own cell proteins and other components. A serial advanced purification technologies such as chromatography are used to ensure the final product is highly pure and free from contaminants. The purified protein is then formulated into a single drug product used for administration. This is how insulin is produced in a large scale industry. We have two vectors over here. One goes for the A chain of the insulin
If you have any questions, please ask her.